Sebastian Wachsmann-Hogiu, and I think he spent a good day in visits. Richard Sandberg has been his student host, and I'm going to ask Richard to introduce them right now.
Sebastian Wachsmann-Hogiu got his bachelor's degree in physics and biophysics at Bucharest University in Romania, and in 2000 he got his PhD in physics from Humboldt University Max Born Institute in Berlin, where he did his PhD work on time-resolved Raman spectroscopy. And then he did a 2-year postdoc in the chemistry department in Pittsburgh at Carnegie Mellon University, and then spent a few years as a research scientist at Cedars-Sinai Medical Center in Los Angeles. In March 2005, he became faculty with the surgery department at Cedars-Sinai and was the director of the Advanced Optical Imaging Laboratory with the Minimally Invasive Surgical Technologies Institute. And so he's had a lot of experience on taking imaging technologies and applying them to the medical industry. And since April 2007, he's been the facilities director at the National Science Foundation Center for Biophotonics Science and Technology at UC Davis. And he was a recently appointed associate professor in the pathology department at UC Davis Medical Center. His research right now is focused on time-resolved Raman spectroscopy and imaging, nonlinear microscopy, and SARS-based biosensors. And he is the co-author of more than 30 peer-reviewed papers and is actively participating in education at CBST and at UC Davis. Thank you for coming.
Thank you very much, Richard. Thank you. And it's really great to be here. It's a great place. I've visited labs and I'm really impressed by what I've seen. Very, very impressive research done here. What I'm going to show is more application-based technologies. It's not so much fundamental research. I think you're more used with the fundamental aspect of research. I'm going to show more applications, or towards applications in the biomedical field. And let me first introduce our Center for Biophotonics. I just spent 5 minutes on this, tell you where I'm coming from and what we are doing at our Center for Biophotonics. The Center for Biophotonics is one of the 15 active NSF centers. We started in 2002. We have, usually these centers run for 10 years. We have 3.5 more years to go. It's an multidisciplinary, multi-institutional center. We have institutions from all over the country. Here are some of our partner institutions. It's the Mills College, it's a women-only college, UC Davis, of course, because we belong to UC Davis, UC San Francisco, University of Texas San Antonio, Alabama A&M, and so on, other institutions across the country. And we, again, we are multi-institutional, but also multidisciplinary. We are trying to combine many disciplines: biology, chemistry, and engineering, physics and engineering. Our location is in Sacramento. This is our building. We occupy about 2/3 of the ground floor of this building. It's a brand new building. We moved in there 3 years ago. Right across the street is the medical center, conveniently located. We have collaborators from the medical center. Again, with the goal to develop and apply our technologies to the biomedical field. The Center for Biophotonics has 3 major theme areas: advanced biological imaging, molecular and cellular biophotonics, and medical biophotonics. The first theme, just a few examples, is high-resolution microscopy. I'm going to talk a little more about this later. And X-ray crystallography, serial crystallography, which aims at determining the structure of proteins without, basically by using single-shot X-rays. The second core area is molecular and cellular biophotonics, where we look to the mechanism of atherosclerosis, developing new gene-based optical probes for in vivo studies, in vivo probes, cellular in vivo probes, and biosensors and assays that I'm going to talk a little bit about. The third field, biomedical or medical biophotonics, involves developing of tools that are used to characterize cells, cancer cells, normal cells, and to develop bioluminescence techniques to study tumor progression, or to detect light, to develop techniques for light-activated treat stroke, sorry, and to do optical biopsy. And optical biopsy is something that I am more concerned about, since I'm in the pathology department. And what we try to do is to use optical-based probes for optical biopsy. So the facility is shown here, again, the building, and this is the layout of the old floor plan. We have 4 major optics that are presented here with the experiments running in each one. The advanced imaging lab, we have the structural illumination microscope. It's for high-resolution microscopy. We have an atomic force microscope and a television system. Nonlinear microscopy lab is imaging using coherent anti-Silk Raman spectroscopy. But we also do microscopy and a project that is not here, time-gated Raman spectroscopy. Single cell and single molecule and cell spectroscopy lab involves time-correlated single photon counting microscopes, a microscope, and a laser trap Raman system that I'm going to detail a little later, a spinning disc confocal microscope. And then we have a laboratory where we develop, it's like a prototype lab where we develop a FLIM, fluorescence lifetime imaging microscopy system for clinical applications, fluorimeters and other devices. I'm going to give you a brief introduction to the field now, really quick. The biophotonics, you will be surprised what really comes into what can be considered biophotonics. And a study done by the UC Davis Graduate School of Medicine estimated there is a market about larger than $50 billion per year. And if you include a lot of non-medical application, like even sterilization, UV sterilization equipment, biosensing devices, or other kind of optical instruments, probes, molecular probes, fiber optic sensors, or even if you move to the therapeutics, photodynamic therapy instrumentation, surgical equipment, endoscopes in the medical diagnostic field, and in vitro diagnostics and imaging. But what we are concerned about is more like these technologies, this in particular microscopies that are listed here, technologies that you all are familiar with. And I'm sure I'm telling you nothing new about any of this. It's just a list here. We are developing sensors and assays. We're concerned about sensors and assays. This is a list with the potential sensors and assays. Of course, not a complete list. And we are mostly dealing with surface-enhanced Raman assays. And biomedical tools, laser ablation. We have a project that involves high power, ultra short high power lasers to do laser ablation, image-guided surgery. Also something that we try to develop. And of course, we try to do translational research, meaning that we try to see how these technologies can be moved and used in the clinic rather than in the lab. Okay, so fundamentals. I think I'm going to skip this because probably, I'm sure everybody knows what photons are and what light is. And different properties of light are also pretty much known. But what I'm going to say now is that when we try to measure one of these 4 properties, absorption, fluorescence, scattering, or Raman measurements, so we are trying to measure either of these properties. And on a Jablonski diagram, we can see how light can interact with the molecules via a one-photon fluorescence absorption and emission. In the excited state, mostly now it's an excited state relaxation process followed by emission. Two-photon absorption involves the simultaneous absorption of two photons with half the energy, and they bridge the electronic gap to create the same fluorescence photon. The second harmonic generation can be produced in the molecules as well. Rayleigh, Raman scattering, coherent anti-Stokes, and infrared. So the bottom line is that the light interacts in a variety of ways, and we can use all these interactions for our purpose. And again, here, one more time, in a different representation, this specific interaction of the electromagnetic field, which you here consider all the way from the X-rays to the terahertz region. And depending on the wavelength, you can probe different properties. You can probe molecular electronic states if you're looking at absorption and fluorescence in the visible range. You can look at conformational motion of molecules if you're looking in the terahertz region. You can look vibrations with infrared spectroscopy if you're looking into mid-infrared or infrared region, or which can be probed also by Raman spectroscopy in the visible region, or you can probe atomic electronic states in the X-rays. And these are just physical representations of the scale of the size. The size of these objects represents really the wavelength of the electromagnetic field. So this is just how what kind of geometries you can apply here for a one-photon fluorescence microscope. You would use an excitation light, use a dichroic mirror that would separate the excitation light from the emitted fluorescence based on the fact that they have different wavelengths. The fluorescence is Stokes shifted, so therefore can be separated via a dichroic mirror. Here you see also the Airy disks for the one-photon wide field excitation. For the confocal, you see the Airy disk significantly improves because you run through a pinhole, which removes the above and below the focal spot, the light that is created above or below the focal spot. In two-photon, you don't need a pinhole because the excitation generates basically fluorescence in a small spot, in a spot where you have enough photon density to create the two-photon absorption process. And the Airy disk is also improved. Then you have second harmonic generation and so on. And here you see the improvements of a confocal compared to wide field fluorescence. And you see the two-photon spot generated versus fluorescence in a QVAT with a dye in two-photon configuration versus just a one-photon excitation. So what we do is to try to identify molecules based on their spectral signatures and to study the interactions between molecules, look at morphological changes, manipulate biological molecules. But in the end, what we want is to correlate with disease, the changes or the biochemical changes, and to characterize disease, if possible, diagnose and do treatment. So this is the bulk of the talk, basically, the developments and applications of biophotonics. And I'm going to go from the molecular level all the way up through the in vivo and through the organism level. So basically, you at the molecular level, what you can look at is, and what we have been we've been busy with, is basically developing biomedical assays. And biosensors and assays, and this is what I'm getting. I mean, you can do other studies at this level, but what I'm going to talk is biosensors and assays. And usual sensors include enzyme-linked immunosorbent assays. It's the ELISA sensors, which are pretty much the gold standard when it's about detecting protein markers or viruses. There are fluorescence antibody assays and evanescent wave biosensors, which are the SPR, surface plasmon resonance-based assays. Biocor, it's one company that developed these assays. And interference-based biosensors, a similar technique with the evanescent wave, but instead of looking at the plasmon resonance, you're looking at changes in the interference pattern when the light is going through through two waveguides. One waveguide is a reference arm. The other waveguide is the waveguide which has the molecules of interest. Optoacoustic assays even have been developed. What I'm going to talk about is, though, it's SIRS-based spare sensors. And SIRS is surface-enhanced Raman spectroscopy. So therefore, I'm going to spend a little bit on to talk about Raman scattering, Raman spectroscopy. Raman spectroscopy is a technique. It's a technique that measures inelastically scattered light on molecules. And what happens is when light interacts with molecules, it is inelastically scattered on chemical bonds. So Raman scattering is a technique that is sensitive to chemical bonds. Depending on the nature of the chemical bond, of the vibration of the chemical bond, the scattered light differ in frequency. Basically, just to give an example, a CC double bond vibration would be at around 1600 wave numbers, while CC single bond will be around 1200 wave numbers. And therefore, since it's specific to chemical bonds, it's a very potentially a technique that could give chemical specificity, and it can be used to identify molecules, to distinguish between molecules. And compared to fluorescent techniques, it does not require labels. All molecules are intrinsically Raman active, more or less Raman active, but they scatter light by a Raman spectroscopy. Fluorescent techniques, on the other hand, use fluorescent labels, unless you use intrinsic fluorophores. But most of the techniques use exogenous fluorophores, which potentially can change the molecule of interest. It can perturb the system a little bit. Therefore, Raman spectroscopy, it has a unique potential here by being chemically specific and label-free technique. Various types of Raman spectroscopy have been developed, and just to mention the most used ones, Stokes Raman spectroscopy measures the scattered light on at lower energies or higher wavelengths, and this is a typical Raman spectrum. Spontaneous anti-Stokes, it's on the higher energy, higher than the laser excitation energy. Then it's resonance Raman spectroscopy, which uses electronic resonance of the molecule to create a stronger signal, hundreds to several orders of magnitude stronger signal than in spontaneous Raman. Due to the Frank-Condon active vibrational modes, due to the vibrational modes that couple with the respective absorption band. There is then coherent anti-Stokes Raman spectroscopy, which is a 4-wave mixing process. It's a nonlinear process. It's a K3 process that uses 2 identical wavelengths. It's in essence a degenerate 4-wave mixing process. And so is coherent Stokes Raman spectroscopy. And this is stimulated Raman spectroscopy. Just to show what happens in the focal spot of the objective where the light is focused, here you would have a spontaneous Raman process. Here you would have coherent anti-Stokes Raman scattering, where you have while the laser excitation propagates through the medium, and again you have the 2 lasers, at least 2 lasers, and the frequencies of those lasers need to match the difference in the frequencies of those lasers, need to match one Raman vibration. And when this happens, you coherently add up signal on the anti-Stokes side, so creating a coherent signal which is called CARS. Here it's basically depicted a SIRS process. It's very naively pictured here. You have a metal particle, and once you come with the excitation light, you would have enhancement due to the electromagnetic field, which I'm going to go into the details a little later. And again, advantages of Raman spectroscopy: you have molecular identity, 3D. It can look at the structural changes, dynamics of molecules, and so on. But it's very weak. It's a very weak process. It's much weaker than fluorescence. So SERS, or surface enhanced Raman spectroscopy, was observed basically as years ago as a process that gives a strong enhancement of the Raman signal due to the presence of when molecules are in nearby rough metallic structures. And metallic structures include silver particles, gold particles, platinum, or other metallic particles. So what happens? You have a strong enhancement, and you can imagine that the enhancement is due to the plasmon resonance. Plasmon resonance is collective oscillations of plasma, of electrons, within the particle. So when you have electrons, maybe I have it here. Yes, sorry, I have it on the next slide. So here you have a metallic sphere, and you have the electromagnetic field that oscillates, and it drives the electrons inside the metal. And due to this charge separation inside the metal, periodic charge separation, you have a very strong electric field that would extend outside evanescently outside the metallic sphere. And this is what creates, in essence, a strong electromagnetic, a strong Raman signal. On the other hand, there are so-called hot spots when the molecules are in between metallic particles. And here is shown what possible configurations you may find. When you find between two metallic particles, but the polarization of the excitation field runs like this, the electric field will not, basically will not create a stronger enhancement than the enhancement given by the metallic particle itself. On the other hand, when you orient the polarization of the laser along the particles, what happens? You create charge, negative charge here and positive charge here. And if this distance becomes very close, then you get a very strong enhancement. The nanoparticles are usually around 50 nanometers, but this distance can be several nanometers, so the enhancement can become very large. But this is just a very simplistic way to explain of what this may happen. There are other potential explanations and ways to understand the SERS mechanism. And in essence, it is believed that two main mechanisms contribute to the enhancement: is the electromagnetic enhancement and the chemical enhancement. Electromagnetic enhancement due to the electric field, chemical enhancement due to the formation of charge transfer complexes between the metal and the molecule itself. So we try to use this phenomenon to detect proteins of interest, like, for example, cancer markers. Of course, we started to look at a known protein, thrombin, and we developed this construct here. We have a glass substrate, silinated, and a gold particle that binds to the substrate. Then we come with an aptamer. Aptamer are oligomers, single-stranded oligomers, and they can be specific for certain proteins. They have specific interactions with proteins. So, and just like antibodies have, but it's a different mechanism. It involves a change in the conformation of the aptamer. But they can be used successfully as binding agents. And so the aptamer, a thiolated aptamer, was added to the gold nanoparticles. And blocking agent, which is mercapto hexanol, the spheres are treated with mercapto hexanol to block non-specific binding of other molecules And then the protein is added. And only the protein that specifically interacts with the aptamer binds, and what we hope is to measure a Raman spectrum of the protein itself due to the enhancement given by the metallic nanoparticle. So what we hope is to measure a Raman spectrum of this protein. And not only this, but also if there is any other protein in the vicinity that gets enhancement by simply being in a hotspot, but not specifically bound, we hope that we can distinguish between the two, between the specifically bound and non-specifically bound, by comparing their Raman spectra, since Raman is rather specific to chemical vibrations. So we tested this hypothesis. And we looked at the following, the first construct that we looked at. It was thrombin binding aptamer. The aptamer itself gives a Raman spectrum because it's close proximity to the metallic particle. It's the green spectrum, and it shows the DNA peaks, specifically peaks. Then mecaptohexanol gives also a certain spectrum. And when we add thrombin, the spectrum looks slightly different. There is some differences, but it's not that easy to recognize anything. So we did the same thing without mercaptohexanol. Mercaptohexanol gives a strong signal itself, so we thought this may be the reason why we cannot see too much from the thrombin itself. And without the mercaptohexanol, we see the orange color here. We see some differences between the spectra of the thrombin and the other components that are near the particle. So this is very encouraging, and right now we're trying to improve this method, even though the reproducibility of the results is not yet as good as we would like. Another way we tried to do it was so the first method was really direct detection. The second way, it was using a Raman probe. So in the same way, we have the metallic particle, we have the aptamer that is bound to the surface, and the aptamer is labeled with methylene blue, which is a fluorescent dye that gives a strong Raman signal in the presence of a metallic particle. So when there is no protein around, the aptamer is bound to the surface. But when a thrombin molecule that is specific for this aptamer binds to the aptamer changes conformation and detaches from the surface. What you would measure then is a change in the Raman intensity given by the methylene blue, by the Raman probe. And here is what we see. We see the Raman intensity of the methylene blue before binding and after binding, so it decreases. And you see here again, it's decreasing if thrombin binds, and this is a different concentration levels of thrombin. And this is a control bovine cerebrum albumin protein that doesn't bind, it doesn't change the signal. By the way, if you have any questions at any point, please ask. It always helps to make it, to improve the participation and to make it more flexible. Okay, so other applications that I wanted to talk is to move towards cells. And I wanted to give two examples here. One, fluorescence microscopy, and one in Raman microscopy, or Raman spectroscopy, better. In fluorescence, I just wanted to highlight this instrument that we developed in collaboration with UC San Francisco and with the company Applied Precision. And this instrument is supposed to break the diffraction limit in what the spatial resolution is concerning. So there are several techniques now, and it's a very hot field that is aimed at improving the spatial resolution of microscopy, of optical microscopy. And there are various other ways you can do it using higher harmonics, of course. Here, we have tried to use regular wavelengths in the visible and to see if it can be possible to improve the spatial resolution using regular optics. And this is by no means the only method. This is called structured illumination microscopy. There are at least two other methods, optical methods, that can be used. It is the STED technique, stimulated emission by depletion, developed by a group in Germany, by Dr. Stefan Hell. And they used a donut mode for the excitation. And the donut mode depletes the sides of the Gaussian, therefore making the excitation much narrower than you would have otherwise. There is also a palm palm technique using photo-switchable molecules, and that it's a technique that takes a very long time, and it uses photo-switchable. It illuminates several molecules at a time and detects individual molecules at a time. And after identifying the centroids of those molecules with a high precision, it depletes those, turns on other molecules, and the process is repeated many times until a full image is created. But this usually takes a long time. Now, this technique here is using moire patterns, which is are observed a lot in photography, in regular photography. Whenever you superimpose two structures, two patterns on top of each other, and you know that a CCD camera is a pixelated detector, and it is a certain pattern. Whenever you superimpose a pattern like anything that has like a shirt with patterns, for example, you would see moire patterns in that image. And those moire patterns have a different frequency, usually lower frequencies that can be measured. The high frequency, the small patterns you cannot observe, but you can measure the low frequency patterns, like you see here. And this is what this instrument does. It takes a grating. It's superimposing this grating with the object in the object image field, and what it does, it takes several images at different rotations and different translating and rotating the grating, and it's able then to reconstruct, after that it can be reconstructed, a high resolution image. And this is some examples. Actin filaments, for example, are much better distinguished fluorescent beads and GFP labeled neuron. So for cells, another Raman spectroscopy and imaging has also been used for cellular to observe cellular processes. And I just wanted to highlight this paper by Catherine Kneip in Nanoletters 1 year ago that uses SIRS as a pH sensor. Basically, when mercaptobenzoic acid is in nearby metallic particles, it gives a strong Raman enhancement. What happens, though, is that mercaptobenzoic acid has one Raman vibration that is whose intensity changes significantly in different pH environments. So this has been exploited, and you can image a cell and see how this Raman peak changes and draw basically a pH map of the cell. And this I found very cool. I wanted to show it to you. But what we have done in our labs with Raman spectroscopy on cells, it was to trap cells. Trap cells with a laser, and trapping, you know how it happens is because of the changes in the refractive index of the cell versus the environment in which the cell is. And due to the momentum conservation law, you always have the particle that is going towards the center of the beam. And you can trap a cell using a laser, for example, 785 nanometers laser, and you can measure a Raman spectrum with the cell with the same laser. And these are a few examples. This is the trapped here it's a bead, I believe. It's a polystyrene bead. Here are red blood cells. This is work done by James Chen and Thomas Hughes at the Center for Biophotonics. So you can see trapped cells and move them into a sort them, basically. And the idea is to measure their Raman spectra and then sort them based on how they look like. And you have various protein DNA markers and protein markers. And using this technique and measuring Raman spectra of cells, Thomas and James have shown that you have different spectra for normal cells versus cancer cells. And while this takes a long time to measure Raman spectrum, in this configuration it takes about 1 minute or so. It's very unlikely that in this way it will be used for sorting. However, there are ways and techniques, for example, via CARS, coherent anti-surface Raman spectroscopy, where this can be sped up, and we're working towards that. I also wanted to show you something that is not related to cells. It's related to human milk. So basically, we did this experiment in which we trapped milk particles. So you can trap basically any dielectric object that is transparent. And milk is basically a colloidal structure with fat particles. And we looked at human milk globules, and we trapped large particles versus small particles, and we saw their fat content, and this is very important for their nutritional component, saturated versus unsaturated fat, their content in this particular fats. And I found this very interesting. I'm going to skip this. Okay. So let me go to the tissue level now here. To the tissue level, whenever you want to apply optical techniques to tissue, you would need to be within so-called this tissue window or optical window, however you want to call it. What it means is that if you're around 1 micrometer, usually between 800 and 1100 nanometers, the penetration depth of the excitation light is much larger than when you use UV or blue or other visible light wavelengths. And this is due to the absorption of various components. And also the scattering decreases when you're going towards near infrared. So this is what we have tried to go towards near infrared. And one technique that is in particular suitable for excitation in near infrared is two-photon fluorescence, excitation of fluorescence. And what I have worked on it was develop a two-photon setup based on wide field excitation. You know, two-photon images are generated by scanning. It's like a confocal microscope where you scan one beam and you generate the image based on scanning with 2 mirrors or any other scanning mechanism. So here I tried to see if it's possible to do wide field two-photon imaging, and as long as you have enough power in your laser, you should be able to focus your beam into a larger spot than a diffraction limited spot, and to create two-photon excited fluorescence from that particular spot. In addition to that, and this basically uses a lens that it's focusing the beam in the back focal aperture of the objective, creating basically a more or less parallel beam after the objective. It's very similar to a TIRF setup, total internal reflection setup. So the reason why I wanted to do this, because I wanted to combine two-photon microscopy with spectral and with fluorescence lifetime imaging in the same time. And first, I recorded an image here to make sure that it's a two-photon excited fluorescence. A laser power curve shows clearly a power dependence typical for a nonlinear excitation. And correction, because the excitation is with a Gaussian beam, a correction has to be performed, and this is how it's been done and the corrected image. And after that, various pulse excitation, various lengths of the excitation pulse were used, showing that femtosecond, of course, works better. And it shows also the 3D, the possibility to record 3D images with 2-photon excitation and compares to 1-photon excitation really well.
What was your sample here?
The sample is an H&E stained tissue sample. It's a breast cancer sample. The idea was just to show that it works well. An H&E stain, usually, it's very stable and highly fluorescent. That was the only reason why I took this. Okay, but the goal was to show that spectral and fluorescence lifetime imaging can be performed on this. In spectral imaging, what it means is that you record images at different wavelengths. Lifetime, fluorescence lifetime means that you record images at a different time interval after the excitation pulse. So they're very similar in what they measure. You will measure in both situations a data cube, a cube that contains a 3D cube. In 2 dimensions, you have your image. The third dimension is either wavelength or time. So the selection method for the spectral imaging is an acousto-optical tunable filter, which is basically an RF frequency-driven filter, transparent crystal that creates acoustic waves inside, a transient grating inside the filter that would change the wavelength, basically, of propagation through the crystal. So it shows here, when you see the blue, you see 2 different structures with 2 different spectra. You see the red, these are red blood cells, and the rest is the matrix here. And you can see that they have different spectra, as well as different fluorescence lifetimes, since this is a longer living species and this is a shorter living species. The spectral range, 520 through 620 to 680, and other technical details are here. But important is that you can then do classification, you can do spectral reconstruction, and you can put together an image that contains spectral information on each pixel. So when you go on each of these pixels, you can tell not only the object, the morphology of the object, but also the spectrum of that object. So, and you can do spectral classification by selecting one specific spectrum and then doing linear discriminant analysis on that spectrum to see what other spectra look the same with that, and then you create a whole map, a whole spectral map. The same you do with fluorescence lifetime imaging. So this is just to show that this was possible. However, in reality, what happens is, this is again the same idea, but what happens is that oftentimes in real tissue is different to distinguish, and the surgeons try to distinguish different components in the tissue based on their color. But the eye is not a quantitative instrument for color in particular. So if you see the same red here as here, but we perceive it differently just because of the background. Same green here as here, but we perceive it differently because it's a different background. So when for a surgeon, for example, when it looks for parathyroid, to distinguish between thyroid and parathyroid, they look almost the same in color. So it would be good to have a quantitative method to distinguish between the two. And this is what spectral imaging does. It's a quantitative method. And you can distinguish between different, here you have pseudo colors helping you to distinguish better different tissue components. It's too bad I don't have the image showing the real sample without the photographic image, because that would be more obvious. Okay, so in tissue, so this is going to be the last part that I'm going to show. I'm going to skip the very last. But I just wanted to go through this. when you want to do in tissue, Raman spectroscopy in particular, you will have the problem of fluorescent background. Raman is a rather weak signal, and fluorescence, even if you use near infrared wavelength excitation, the fluorescent signal is still overwhelming the Raman signal. Therefore, you need to find ways to avoid this. There are algorithms where you can subtract the fluorescence background, but those are not always very reliable. You can bleach your fluorescence background, but this is not very good if you do in vivo. So one way that can be distinguished is based on the fact that the Raman scattering is almost instantaneous if you excite with a very short pulse laser, and the fluorescence decays over several nanoseconds. So you can do you can distinguish between the two based if you develop a temporal gate. And again, you would do very well, you would measure very well the Raman signal if your gate is very short. And there are several techniques that are aimed at time gating, not in particular for Raman, but the most used ones are listed here: time-correlated single photon counting, frequency domain upconversion, all very difficult techniques. So what I'm focusing now on is the Kerr shutter. And previously, Kerr shutter has been used for separating the Raman and the fluorescence spectra. However, it uses regenerative amplifiers and high energies per pulse, which is kind of damaging or difficult to do such measurements in tissue. So I was trying to develop techniques that are using regular, like oscillators, like the chameleon titanium sapphire oscillators to do this. But let me go quickly over the Kerr shutter principle. When you have your signal here, you run it through a polarizer, you polarize the light. It goes through a Kerr medium, and if you put the analyzer perpendicular on the polarizer, so basically nothing goes through if your polarizers are good. So, but when you come with a fast, ultra-fast laser pulse, you create in the Kerr medium birefringence. And if the polarization of the Kerr laser is oriented 45 degrees with respect to with your signal, then you can get for a very short time a shift in a 90 degrees rotation of the polarization, which is basically a ultra-fast half wave plate, if you want. And this is what you're doing by the wave plate. The half wave plate would last only as long as the medium, as long as the laser pulse is in. And for that period, the light will go through the analyzer with the detector, but after that, the shutter closes, and the fluorescence that comes later will not go through. So we have looked at several configurations, and one is basically using a nonlinear medium such as CS2 or titanium dioxide. We have the signal with the Raman and the fluorescence. We have the laser, the Kerr laser, and we run them together. And in this configuration, you need high energies per pulse. What we have tried also is to run it through a photonic crystal fiber, which has, even for short distances, due to the fact that it's a very narrow core, you can reach very high densities, power densities. And eventually you could create, we are currently investigating this configuration in an interferometric setup as well as in a Kerr shutter setup. And this is the optical design. I'm not going, it's basically the same, and this is what we have done in the lab. Okay, and I think that would be all for, I would have had a little more for the in vivo, but I think that's enough for now. Okay, so in vivo, I just wanted to show quickly some of our, from the time I was still at Cedars-Sinai in Los Angeles. We developed a in vivo imaging box, basically, that looks at mice and it looks, of course, you have many advantages to do this. You don't need to sacrifice the mice. You can do toxicological research. You can do cancer research, pharmacology, and it compares well with. It has advantages and disadvantages compared to other imaging techniques that are usually employed for small animals. But what we can do is to combine it with other techniques, fluorescence lifetime imaging, or fluorescence, or hyperspectral, meaning spectral imaging, like the one that I described earlier. And just to give a quick example. Oh, here. So if you use a drug carrier of this sort that has a drug attached to it, I'm not going to talk about the chemistry, just the fact that you have a fluorescent tag, and if you inject it in the tail vein of the mouse, it goes into the mouse, and you can follow where this construct goes. You can see the veins, you can see the mouse illuminating, really nice. And there is a tumor here, and you can do pharmacokinetic studies, how fast this drug goes to the tumor, how well is retained by the tumor, and basically the specificity for the tumor. And that's basically it. These are the people that have contributed and have worked with me on these projects. People from Center for Biophotonics, the center director, Dennis Matthews, Thomas Huser, Steve Lane, my postdoc, and my students here, Diana Thompson and Florian Knorr, working on the time gating technique. People from Livermore we work with for the CERS project, and people from the Cedars-Sinai Medical Center I have worked in the past. And thank you very much for your attention. So if you have questions.
You had that instrument which was doing the multiphoton with the spectral and time information. I'm not a doctor or anything, but that seems like it could, for tissue, like there's a lot of information in that. Or do they have specific examples of, we think we can diagnose this, that, or the other with this technique? Or?
Yes, so it has a lot of potential. There are technical issues, though. So it's never looking that easy. So first of all, the field of view is rather small. You have about 50 micrometers that you can look at a time. So in vivo, for in vivo, this is very small. Yeah. So the imaging techniques that I showed you, the whole animal, it's more telling than what you would have with a small field of view that you would look with this 2-photon, wide field 2-photon. But indeed, it has potentially a lot of advantages if you know where to look. And you can, for example, and this was the hope also, that you go with let's imagine now. Yeah, we go a little bit and imagine what would be. So if you have an endoscope-based device, and you go with an endoscope inside in the lung, for example, and you want to, in the stomach, and if you want to. You look with the endoscope, you see something that looks suspicious. You can go then and zoom into with this device. Yeah, so this would be one possible application. But I think that is still a long road there.
You mentioned that CARS wasn't a very sensitive technique, but then you showed that for CERS, there's 12 to 14 orders of magnitude increase the sensitivity theoretically. That would make it a very sensitive technique. Has anybody actually seen those kinds of increases experimentally, or is that still a dream?
It is not a dream. It has been observed in certain situations, even 15 orders of magnitude increase. So, and single molecule studies can be done in this way, and they have been done. So, what is more, and these are isolated cases rather, when the molecules are in a hot spot that is very suitable for this enhancement. But this sort of devices are not very useful. First, because the signal is not very reproducible. You don't know how to place your molecule always in the same hot spot. So the concern is rather to develop or to nanofabricate particles that give you the same enhancement, even though maybe just 7 orders of magnitude, but they give you reproducible enhancement rather than extremely high enhancements. I think this is an area that is a lot of development currently, to fabricate substrate that have that give reproducible results. So regarding cars it is sensitive. It gives 3 to 4 orders of magnitude stronger signal than the spontaneous Raman. But it doesn't compare with SIRS though. SIRS has the disadvantage that is you have metallic probes. Cars is fully label-free. That would be the advantage of cars versus SIRS, even though the enhancement is lower than SIRS.
Can you use your time-gated system to image deeper tissue?
Yes, very good question. So actually, I'm very fond of this technique. Yeah. So I have put a lot of effort into this recently in the past few months, because I believe that it has a lot of potential, not only for separating the Raman from fluorescence, but also looking deeper into tissue by choosing your gate in such a way that you can look at different depths. And of course, your time resolution will limit your sectioning ability, but it is possible to look. Not yet. We're still developing the system. Maybe I wasn't clear enough. So we are trying to, this technique has been developed in the past and has been used for Raman, but at extremely high, what I call extremely high energies per pulse in the range of microjoules to, or tens of microjoules per pulse. So any of these energies would be very unsuitable for biological applications. So what I'm trying to do is to make it so that it can work at nanojoules level. This is the new part that we are trying to do. Thank you.
We have pizza in Benson 145. Please join us for that, and Dr. Martinez will be there as well.
Yeah, I can answer more questions if you have there. Thank you. So this is yours.